Good morning and welcome to the International Plant Breeding Seminar. Today is Thursday, April 6th. We're about a minute ahead of the time. Today we have the honor having Dr. Luc Jannink from Cornell University. He's an adjunct professor at the School of Integrative Plant Science, Plant Breeding, and Genetics. And he's also a research geneticist at the USDA ARS Robert Holly Center for Agriculture and Health. And the primary focus of Jean-Luc's research is on developing statistical methods to use DNA markers in support of plant breeding programs. Thank you so much, Jean-Luc, for accepting the invitation to speak to us. It's a pleasure to have you as part of this series. And I'm going to stop sharing here and give you the floor. I want to remind the audience that it would be nice if you shout out from where you are joining the seminar. Put that in the chat, and also reminded you that the questions should be posted in the chat, and it will be referred to Jean-Luc at the end of the presentation.
Okay, thanks for the introduction, Carlos. And that was a pretty cool video. I really appreciated all of the
I specifically picked the music because of you.
Yeah, that was good. That was good. Hang on, let me just see where we are here. Okay, so we're ready to roll?
Yes, we are.
Okay. Thanks for the invite. So what I'm going to be talking about is the road to genomic prediction in cassava. Right, I'm Luc Jannink, I work for USDA, and I'm adjunct at Cornell. Okay, so I have a bit of a preamble, and then I'll launch into the along to the talk. The first thing I want to say is very much next gen cassava, not necessarily what would have to happen in cassava. I think there are multiple different ways of beginning or introducing genomic prediction into breeding programs. And here's one. And I think we were relatively early on in this whole process, so we made some mistakes that I'll share with you, and that way you won't have to make those mistakes, right? So just a brief about Next Gen Cassava. It's a huge project. It's really sort of astounding that way. I think if you consider Hawaii to be part of Oceania, then we cover 5 continents. The most important thing of this slide really is our vision of empowering farmers through innovative cassava breeding. Yeah, so just another way of showing how big we are is the list of people who I feel have contributed to some of the aspects that I'm sharing with you here. Here's me as part of the Cornell University contingents working for USDA, but you can see that I'm a small part of a very large project. So here's my assumption about the people who are listening and have tuned in. Really, Next Gen Cassava is here because there are smallholder farmers who can get better opportunities from better varieties. And I'm assuming that you're watching because you also want to offer those kinds of opportunities to people. And I hope that by illustrating some of the work that we've done with Next Gen in bringing genomic prediction into cassava breeding, that you'll be able to take those and work with them. They're not all technical. Some of them are organizational. So, okay, just in case you're not familiar with cassava, there it is. It's a starchy roots crop. Starch has a lot of calories, and cassava is an important source for millions of people. Furthermore, it's grown a lot by smallholder farmers. This is because it is a resilient crop and can grow on low fertility lands, right? This starchy root, it doesn't have a whole lot of protein, so not a whole lot of nitrogen fertility is required in the soil. As a root crop, as opposed to a grain crop, it doesn't have a critical pollination period. So it has this mechanism that leads it to be more stable, and therefore it's an important food security crop. Just from the point of genetics, it's outcrossing and clonally propagated. So that's of course relevant to how you go about using it in a breeding program. So this is sort of the outset of the project. Cassava yields in Africa have been increasing over time, about 1% per year over the past 5 decades. That is not fast enough. And when cassava became a priority crop for the Gates Foundation, they were looking for a way to bend that yield curve upwards. Remember that this 1% per year, that's not all genetic. In fact, maybe none of it is genetic. We really want to accelerate the genetic contribution to increasing cassava yields in Africa. So genomic prediction, just very briefly what that is. It's a way to accelerate the breeding cycle. You start with a so-called training population. These are individuals that have both phenotypes and genotypes. You've invested in genotyping them and put the work into collecting phenotypes. You use this data to train a statistical model. It's a prediction model. It's going to enable you to predict an expected phenotype or the phenotype of offspring from genotypes. So then you bring along new breeding material that you've genotyped, and you use the statistical model to calculate genomic estimated breeding values. So it's a breeding value. This means that it's a prediction for the performance of offspring, right? It's the ideal selection criterion. So you use that to make selections and then you move on. And again, because genotyping can be done very early on in the life of an individual, and so you don't spend the time increasing the crop and evaluating it phenotypically, you can greatly accelerate the breeding cycle. Now, coming into this next-gen cassava project, I had a view of genomic selection that's similar to the view of New Yorkers in their view of the world, okay? So this is a map of the world according to New Yorkers, and you can see that 80% of it is taken up by New York City, right? And I had a similar kind of, perspective, I guess, bias on the importance of genomic selection within the scheme of producing varieties for farmers. But really, there's a lot more to it. And, a little bit of what I want to talk about has to do with the extra stuff. But I just want to start by saying that, okay, so you need a lot more than just this genomic prediction. You need to be able to make crosses in the crop, which for anybody working with cassava, that's not trivial. You need careful data management. Okay, so collecting all of the beautiful data in the world won't help you if it gets disorganized. You need germplasm exchange. Cassava was domesticated in the Amazon basin. We are targeting Africa. We need to bring new genetics into Africa, particularly to address diseases. You need a careful sense of how you're going to move your breeding material along, both how you're going to design your breeding goals and how you're going to decide when a particular clone, in our case, should move from one stage to the next. Probably one of the most important things is deciding on the breeding goals themselves. Some of these breeding goals are self-evident. So in Eastern Africa, cassava is affected by cassava brown streak virus, and clearly we need to increase resistance to that virus in cassava. Some of it was less obvious, and I think we were somewhat unaware of the importance of product quality early on. And we didn't have good ways of measuring product quality. So that's been a very important aspect of the Next Gen project going forward. But if you don't have the right breeding goals, then breeding slowly is better than breeding fast, right? So genomic selection in service of the wrong breeding goals is actually a bad thing. And then finally, I'm going to make an emphasis on quality management. This is sort of the wheel of quality management. And yeah, collecting a whole lot of data isn't useful if it's not high quality data. So you can see this is genomic selection in the grand scheme, right? It's sort of buried by many other aspects of the breeding scheme that are important. That said, I don't think it's completely unimportant, and it is what I'm going to talk about mostly. So yeah, I'm going to talk about the trained population that we started out with, the marker system, and the breeding scheme that we used, that we were using these predictions for. And I do have some alternative suggestions, possibly things that would have helped us out, but just things that you ought to consider if you're moving towards, the road of using genomic prediction in your own crop. We had some surprises as we got going. I think that's not in and of itself surprising. What's important then is how we respond to those surprises. So talk a little bit about that. And then, of course, we're proud of our project, and I want to show some outcomes that illustrate that. Okay, so this is where we started. This is sort of the variety development pipeline, or VDP, for IITA at the outset with phenotypic selection. It involves a minimum of 5 years for the breeding cycle itself. So going from cross to evaluation to selection to crossing, and a minimum of 8 years before you would have from crossing to variety release. So this is what's commonly sort of represented as a triangle, this VDP. And so the genetic gain population that was our initial training population came from IITA maintaining clones that had gotten sort of the farthest in their breeding pipeline, in their VDP. So these are all very high performing clones, and basically year after year, they would collect these clones from their most advanced trials, right, the tip of the triangle, and they would hang on to them. So over time, we had this large collection of, very good clones that had high quality phenotypic data associated with them. So this is the genetic gain population, and it was our initial training population. And I should say it's an incredible luxury to have this as a training population for a clonally propagated crop, right? It's expensive to maintain clones, but IATA had done that. And so we had this instantaneous training population where we could just extract tissue from and extract DNA from all of these clones that they'd maintained. And we had the phenotypes associated with them. And all of a sudden we could move forward with making predictions. Now, so that was a big pro, lots of high quality phenotypic data. There are some possible cons to this training population. The clones are highly selected, and I'm not entirely sure that that's a bad thing, although I'm in hindsight, well, you'll see. What does mean that the mean of this training population is pretty far from the average population mean because they're highly selected. And I do want to say that among highly selected individuals, negative correlations can arise among favorable traits. So just to illustrate this, I hope you can see this. Imagine two traits. The concentric circles sort of indicate that they're uncorrelated at the outset. But now we're going to select towards high values for both traits, right? We're going to keep the individuals that are high for trait 1 and trait 2, so the red individuals, and discard the blue individuals. And so then what you're left with is a kind of wedge like this. And you can see that this wedge has a negative correlation. So, and we know this in practice, that actually, for example, in cassava, the correlation between dry matter content and fresh root yield is more negative in the advanced stages of trials than in the preliminary stages. So this is a characteristic now of our training population. And it is a characteristic that is, it's hard. I don't know, to be honest, how much it's affected us, but I think you'd be safer if you can collect individuals that are earlier on in the pipeline. And, if you're starting out and you haven't had the luxury of hanging on to these advanced individuals, that's probably where you'll start anyway. So remember that this cassava project, it started over a decade ago. At the time, genotyping by sequencing was the state of the art for sort of low cost, high density marker systems. So this is just a cartoon illustrating the difference between genotyping by sequencing versus whole genome sequencing. In GBS, you just sequence at the site of restriction enzyme cut sites. So you have a reduced representation of the genome. You don't have to sequence the whole thing. And we had an in-house protocol for this. The procedure itself was very much developed by Ed Buckler, who's been part of the project. So we could do it. It doesn't require a whole lot of genomic resources up front. So that's what we did. I think nowadays you would use a different approach. And so this is a slide with information that I've pulled from Breeding Insights. Thanks to Moira Shaheen for providing this to me. Breeding Insight is a project that's funded by USDA that is to help specialty crop and livestock breeding programs within USDA ARS to bring new technologies, both genomic technologies, excuse me, and phenomic technologies into their breeding program. So imagine you have a crop with no genomic resources at the outset. It's quite useful to have a reference genome, surprisingly cheap to make that nowadays. Then you want to do marker discovery, so sequencing to identify polymorphisms, and then develop a robust assay from these polymorphisms. So that costs, $50,000, give or take. certainly not trivial, but given that you're starting with 0, it's not that bad. and then the beauty of it is then you have a genotyping platform that is very robust, that'll give you about 3,000 markers for $10 a sample. So then this is all outsourced, right? Initially with NextGen, we were working in-house, developing the markers in-house. Outsourcing is just so much easier and allows the breeder to focus on breeding. So we work with Intertech and Diversity Arrays Technology. They are companies that that Excellence in Breeding has been working with. And so, you know, what we did with GBS doesn't make sense nowadays. This is the breeding scheme that we jumped into. We really went all in. We wanted to maximize the advantage of the breeding cycle time shortening that genomic prediction can provide. So basically for 1/2 of the year, we would be growing seedlings, extracting, sampling tissue, doing the DNA markers, making predictions and selecting. And then the other 1/2 of the year, they would be cloned and sent to a crossing nursery. We have subsequently abandoned this approach. We're taking more time to look at the new clones entering into the pipeline. So here's depictions of different timing lengths for genomic prediction. Like I said, we started with a 1-year breeding cycle so that individuals would transit from the crossing nursery to the beginning of the variety development pipeline and cycle through in 1 year. You have then no phenotypic observations on the individuals that you're selecting. And to some extent, they're subject to natural selection, which may work against breeding objectives. And I'll describe that down the road. You know, if you take 2 years, now all of a sudden, you have some brief observations or single plant observations of your new clones. And in particular, at least for the specific aspect of natural selection that has plagued cassava, it'll allow you to eliminate that. And now frequently, actually, we're waiting 3 years. That gives us our first clonal evaluations. So we have actually plot level yield data on these individuals before we select them. So these are just things to think about as you're entering. So you might ask if you have to wait until you have okay observations on all these clones, what's the advantage of genomic selection? Why bother? Okay, so first of all, you're still cutting the breeding cycle time from 5 years to 3 years. Genomic prediction focuses on breeding value, right? As opposed to what you get from phenotyping, which is the genotypic value. So the breeding value, the genotypic value is the performance of the individual itself. The breeding value is the expected performance of its offspring. So that's the more pertinent, I think, selection criterion. Most important thing, though, I think, is that genomic prediction allows information sharing across all of the individuals that you're phenotyping. So individuals that are related to each other, they provide information that enables you to make better predictions for each individual. And I just want to note that I'm saying these are advantages of genomic prediction, and genomic prediction provides them in full, but you can also get all of these benefits from a pedigree-based relationship calculation. So, you know, if genotyping is just not a possibility for you at this point, you know, do a good job of recording pedigrees and integrating pedigree-based relationships into your breeding. I would say genomic selection, it took off almost immediately in livestock breeding, precisely because everything that they'd been doing up till then relied on pedigree relationships. And so if you do a good job of working with pedigrees, you're setting yourself up beautifully to work with genomic prediction. I mentioned that we jumped right into a 1-year breeding cycle, and I also just want to mention something that of a sort of easier approach to start genomic prediction, which is predicting within family, where you test half the family and you predict half the family. So dark green is phenotypic testing, light green is only doing genotyping and then prediction. You're at some point now you're doing you're phenotypically testing the whole family. You can transition to reducing the amount of phenotyping that you're doing and predicting another half. So this has a very low barrier to entry. You don't really need to change the procedures that you're using in your breeding program. Within family prediction is highly accurate, and it's allowing you to build up over time a large training population, because all of these individuals now, they have marker data, right? And so you're accumulating this across families, across breeding cycles. So that can be then a powerful training population down the road. The cons is that indeed, you're not changing your overall breeding scheme, so you're not reducing the generation interval. And it does require relatively large full sig families. Just a second kind of advantage is the previous slide I showed evaluation in one environment. You can, once you have the genotypes, distribute the family across 2 or more environments. So here I'm imagining an optimal environment in drought 1. I'm splitting the individuals that I'm evaluating. Because of the genotyping, I can do reciprocal prediction so that I have an estimate now of the performance of all of the family in multiple environments. Okay. So there's some thoughts on the training population and getting started. Now I'm going to launch into sort of what happened, like what are the outcomes of our attempts at genomic prediction, and just to upfront say that we are experts, and an expert is someone who's made more mistakes in an area than anyone else. Okay, so yeah, I think we were out front, and being out front means that you take turns that maybe you shouldn't have. You have to be willing to learn from your mistakes, so you have to be willing to look at them and probably share them with others. And the advantage of sharing them is it helps you learn from them, and also it helps others learn from them. Okay, so what happened? So at the very first, we actually didn't get any gain, and at the beginning, I was pretty nervous about this, and I think everybody involved in the project was wondering what was going on. We hung on, and by the second cycle, so gain had sort of begun, and I think the explanation for this is fairly straightforward, actually. Remember, the source of our training population is this genetic gain population, which is the tips of the triangles, whereas the first cycles, we were looking at the performance of just a number of the clones that were entering into the variety development pipeline. So again, the population mean of this genetic gain was far above what you would expect it to be at the beginning of the pipeline. So here we're looking at individuals that are at the beginning. These were individuals at the end. I think what's important here is that there's gain going from cycle 1 to cycle 2 that are being evaluated sort of at the same stage. Another thing that we realized fairly early on, I mean, I didn't realize this, but I think it turns out to be true, is that we selected for early branching. This 1-year cycle meant that we could only cross with clones that were flowering early. But flowers occur at the site of branches in cassava. And so all of our crosses were being done with early branching clones. So you can imagine what that does, right? And this is the natural selection that I was referring to. There was natural selection for early maturity, which meant natural selection for branching in cassava. Fortunately, we also have a component of the project that is looking at non-genetic ways of inducing early flowering. And that is sort of working pretty well now. We didn't have it available to us at the beginning of the project. It is available now. And so that offers the possibility of taking clones that are late branching, that are farmer preferred in that way, but inducing them to flower so you can nevertheless breed with them. Okay, so this is the slide that is sort of the hardest for me to present and possibly the most embarrassing, but I think it's one of the most important slides. We discovered a much too high rate of genotype mislabeling. So this was all the way into year 4 of the first phase. So what do I mean here? I mean that you start working with individuals and you're genotyping them, and of course you genotype some of them more than once. And so here in this column, I have individuals that were genotyped twice, and in this column, individuals that were genotyped 3 times. Here's an example of what that looks like. So this, 11206 clone, it was genotyped multiple times, even if there were slightly different names. This is the library number. And then we had 4 different institutions, 4 different breeding programs that we're working with in Africa. And you can see that the frequency with which you'd be looking at 2 individuals that were labeled the same, they should be the same, but the genotypes were telling you that they were not the same. That happened 20% of the time in this case, and so on. That frequency of mislabeling was just much too high. This was a time of reckoning. I would say this is what happened to us. Initially in the project, we had a concern with the quality of historical data. We worked on that, but then we had this major effort, the crush of trying to get our project up and running, of trying to run through these breeding cycles and so on, so that we just sort of assumed that everything was working well. And then at a certain point, we realized, we better check this. And then to our credit, and this has to do with the response to discovering what I just showed you, we implemented systematically and not just across the genotyping effort, but efforts to improve quality control. So what do I mean by that? We already had Cassava Base as a data management tool, but we really tried to set it up now as a quality management tool also. I'll describe that. And then we wanted to motivate all participants to use these tools. So we developed this Quality Champions program. Okay, so here's Cassava Base, this idea of a digital ecosystem. What's important here is simply that there's no exiting, there's no transcribing of information from paper to computer and back again. So all of the data stays within sort of identity tracked data sets, spreadsheets and databases. That's one component. The other component that's critical is that anything that's out in the field now has a barcode, so that there is a computer way of identifying and tracking individuals. I mean, there's a reason why barcodes, the uptake in the industry of barcodes was tremendously fast, and nothing happens in industry without a barcode. They're just effective at enabling digital identification of things. And like I said, we developed a quality management approach, our Quality Champions program. It's collaborative, so we have received help from Excellence in Breeding and from the breeding program assessment team, BPAT. It's iterative. The quality champs, they meet regularly throughout the year. It's facilitated by digital technology. We've tried to move to being proactive as opposed to just reacting to the fact of, we have a lot of mislabeling. We have this advantage of size where we have these 4 breeding programs, and there are more actually, because there's a, CCOP, what does it stand for? The partnerships, the community of practice partnerships. So we have the opportunity to learn across programs. If one program does something well, what can the others learn from it? And I think an important part of this Q-Champs program is that it's not really top down. It's really the people who are out in the field, who are doing the work, who are being motivated to figure out ways to improve quality control. So all of that has led to some success. So here's a slide with the genetic trend over time with separating out the pre-next gen era and the genomic selection, or what we're calling the next gen era. There's a little gap in between where we removed data because there had been crossing between white clones and high carotenoid yellow clones. But anyway, the point is, I think that here we have a serious slope of gain from selection. This is gain of an index across yield, dry matter, and for West Africa cassava mosaic disease resistance. It still doesn't include these really important food quality traits. So that's something that we're learning that we have to add into this. But I think any plant breeder would be terribly proud of a slope like this, and we are. And that's led to some variety releases. So again, we're hopeful that we're having an impact at the farmer level. These are releases that have specific advantages in terms of their food quality. All right, so it's time to start wrapping up here. Delivered that faster than I expected. So really, I mean, genomic prediction is important, but it has to work in the context of high quality data. And, we really discovered some problems with quality as a result of using DNA markers. But whether you're using markers or not, now is really the time to put your house in order. And if you like us, wait until you have markers to prove it to you, it can be painful. In the Next Gen Cassava project, I think we really jumped into some of the more challenging ways of using genomic prediction in a breeding scheme. I remember talking to some colleagues from private industry and describing what we were doing, and they were like, "Ah, wow, good luck." And I think it was because they had some experience with this, and they were realizing just how much we were taking on, and we were a little bit naive. So at the outset, think about easier approaches. One way to make things easier is to find partners and outsource. I mentioned Intertech and diversity raised technology. I don't think any breeding program really should be doing DNA marker work themselves at this point. It's just easier to outsource. Companies can do it better than you can, and it takes a load off your mind. I should say too that the Next Gen Cassava project was very fortunate in that we were doing our thing at the same time as excellence in breeding was really taking off, and this the BPATs, the breeding program assessment team, was out there. So we had the advantage of a number of collaborators and help. So take advantage of that. I think for clonal crops, assembling a trained population is just really a major task. And make no mistake, a large training population is really important. I think there's plenty of papers out there in the literature now with populations of 200, 300 doing genomic prediction. I think for serious breeding, that's not adequate. Clones are expensive to keep around. So if you have good phenotypes, at the very least, sample tissue and dry it down and try to store that until you're ready to extract DNA from it, or you can just have the DNA extracted, again, outsourced, and stored until you're ready to do the genotyping. And all this is to say that I think, running a breeding scheme is relatively easy, right? Going through all of the motions is easy. what's really hard is identifying the right breeding goals, doing it efficiently with quality data to ensure that your output is adopted. And that's actually really hard. And so that's what one of the things that Next Gen Cassava has taught me is just how much more there is to a breeding program than effective prediction. So with that, I'd just like to thank the many partners who have contributed to this. It has been and continues to be an amazing learning experience being part of this community. Thanks. So I guess I can take questions.
Thank you, Jean-Luc. Thank you so much. You want to stop sharing, probably?
Yeah, let's see. Oh, right.
Okay, well, we already have a couple of questions, okay?
All right.
Okay, very nice perspective, Dr. Jannink. I was wondering about how many markers in general are used for genomic selection in cassava.
Yeah, that is a good question. And there's some contention. I mean, you can get pretty decent predictions often with small numbers of markers. So just in the few hundreds, for example. I think that's a bad idea. So we actually, we use this DART-seq LD genotyping at this point, but then we also impute. So I think a lower-ish density, 2,000-3,000 is okay, as long as you're imputing to a higher density. And so we have, I mean, for us, imputation has two aspects. One is getting to higher density. The other aspect is, we started our whole project on genotyping by sequencing. Then genotyping by sequencing was patented and we didn't have access to it anymore. But a lot of our historical data then is tied to genotyping by sequencing. So that's one of our reasons for imputing, in addition to wanting to have higher density markers. So anyway, the direct answer to the question is, I think a few thousand is adequate if you're imputing to higher density. If you want to not do imputing for something like cassava that's outcrossing, I mean, I'm not sure who all would agree with me on this, but I'd be most comfortable with, in the tens of thousands, say 20,000 markers. You can get, for a short time, you can get prediction accuracies that look decent with much lower densities. I think in the long run, you're better off with higher densities.
Thank you, Jean-Luc. Again, a reminder to post your questions in the chat. We have another question. Okay. Thanks so much for an excellent presentation. In perennial crops, the majority of breeders still rely on molecular-assisted selection. Can you please explain when will be a good time to move from mass to genomic selection, and pros and cons?
Yeah, good question. Well, okay, so marker-assisted selection, what you've done there is you've identified a known association between a marker and a large effect QTL. And the reason I say large effect QTL is because if it wasn't a large effect QTL, you wouldn't be able to establish that association, right? But we know that many traits that we're interested in are determined by many loci of small effects. Okay, so the classic case, right? I mean, the model system quantitative trait in humans is height, human height. They've done a GWAS now of human height with over 5 million people. There are over 7,000 independent loci that affect human height. That we know this statistically. Okay, so and that's an underestimate. The larger the GWAS gets, the more loci they find. So there's no doubt that for the traits that we care about, there are thousands of loci that contribute to this. And you can capture the effect of those loci with genomic prediction models in ways that you just can't with marker-assisted selection. So when is the right time to transit? Yeah, I mean, I think it really has to do with, I mean, you have to have a breeding program that's large enough so that the genotyping doesn't take up, I mean, an undue amount of your budget. At the end of the day, the thing that drives breeding forwards is high quality phenotypes. The genotypes enable you to get more gain out of good phenotypes, but if you don't have the good phenotypes, you're nowhere. So yeah, it's a complicated thing. It depends on the size of your breeding program. But you know, if you have a good size breeding program, the time is now. I mean, you're going to be able to work with quantitative traits, make predictions, accelerate the breeding cycle. Yeah, it's worth it.
Another question here is, do you have a threshold as to the number of genotypes that constitute adequate train set in cassava?
That is a good question. I think there's, yes, well, it's always the larger the better. We started with 700 in the IATA breeding program, and you know, around 600 in most of the other breeding programs. So, okay, a real Achilles heel of genomic prediction potentially is loss of genetic diversity. So when you start, there are two aspects to this. When you start breeding with prediction, you accelerate the breeding cycle. Each selection event is a bottleneck, right? So you're generating more bottlenecks per unit time. The other thing about this is genomic prediction is working on genetic relationships. And so that means that with genomic prediction, you're doing more selecting among families and less selecting within families than with mass selection. Okay, but when I say mass, I don't mean marker assisted selection. I mean, you know, just phenotypic selection. Selecting among families versus within families leads to higher drift, lower effective population size. The larger your training population, the less that is true. The larger your training population, the better, more effective you are at within family selection. So you might get some good gain with a small training population early on, but it's going to be at the cost of drift. So it's, you know, I think if you have good ways of maintaining your genetic diversity, you can probably handle a smaller training population. But yeah, I wouldn't want to enter into it with smaller training populations than what we had at the outset of the Next Gen project.
Good. Another question or two here together. How to develop a higher density marker, and do we need to know the location of the genes, or is it a genome-wide marker?
So I'm a true quantitative geneticist. I think that these effects are many loci distributed all across the genome. So just genome-wide markers are what you need, and whether they are in close proximity to genes or not doesn't matter too much. Yeah, I mentioned the pipeline that Breeding Insight uses for marker development. I think that's a fine approach. Yeah, first developing a reference genome, about $5,000, then taking important genetic diversity that is important to your breeding program, not genetic diversity globally, but important diversity for your breeding program, and doing a good job of sequencing that diversity, and then extracting markers from there. So yeah, I think that's a good approach. I see a question from Stuart Gardner, my good friend.
Yeah. So yeah, and the two next questions sort of related. Did you identify any of the causes of the genotyping errors? And there's another question below. It says, how did you know there was an error in genotype?
Okay, so maybe I was not entirely clear about what I was presenting. so this is what what I was trying to present was genotype mislabeling. In other words, two individuals were labeled the same thing, so that if you went by the label, you would pool data from those two individuals, right? They were labeled the same thing, but the genotype showed that they were two different individuals. So it's not a genotyping error in the sense of, this locus should have been a heterozygote and you scored it as a homozygote or something like that. So I think in clonally propagated crops, genotype mislabeling, it just arises over time, over clonal propagations, right? Just think, when you go through a breeding cycle, there are at least 10 different points when, you have to move a label somehow. You're going from seed to planting, you're going from planting to putting a plot stake in there, you're labeling a harvest bag, et cetera, et cetera. Now imagine that each of these steps, you have a 0.5% error rate. 0.5%, I don't know if that sounds like a lot to you. It's very small. I mean, I think humans have difficulty estimating how often they make mistakes. You think of a procedure that's easy, you think, oh, it's impossible to make a mistake here. But if you do it 200 times, you're going to make one mistake. But then you do that 10 times and your 0.5% is 5%, right? And then you do that 3 years and your 5% is 15%. It just accumulates over time. So there's no shame in it. It's human, and so you want to use barcodes and stuff like that to seek to avoid it.
Mm-hmm. Despite genomic predictions, more, why do you still have a slow increase in yield gain for cassava in West Africa?
Okay, well, I mean, so the graph I showed you was 5 decades worth. Our next gen cassava project has been going for about a decade. But I mean, there's lag time. I mean, the first 5-6 years was easily just trying to get it to see if it was going to work. So my hope is that if you ask me this same question in 10 years, that the yield curve will be bent upwards. I think it's happening.
Good. What steps have you used or plan to use to ensure genetic diversity is maintained in cassava?
That is also a good question. So one of the things that we've done is just, and this was actually really Ismail Rabi's idea, cassava does suffer from inbreeding depression. When we make these crosses, we want to avoid crossing individuals that are at all related. So one of the things that we did was take all of our selected individuals and cluster them, and then primarily make crosses between clusters as opposed to within clusters. So that avoids some inbreeding depression. Now, maintaining genetic diversity, that you need to do more than just that. And I guess the state of the art for us right now is what we are calling genomic mate selection, where we are not just selecting the best individuals, but we're trying to identify which crosses to make between individuals. And so when you do that, you're using the genetics also to decide which two individuals are probably most widely separated, and therefore it makes sense to cross them. Something that I think we should do more of is this approach called optimal contributions. So here in optimal contributions selection, you're trying to select the overall group, not just the specific individuals. And the value of a particular individual arises not just from its predicted breeding value, but from how genetically unique it is relative to the remaining individuals that are being selected. So you're essentially you're selecting for high response with a penalty against inbreeding. They're well worked out quantitative genetic methods for that. Yeah.
Just one more question that just arrived. Are there different ploidy levels in cassava? If there are, does this complicate the breeding management toward the goal of developing half-seed family?
You know, that's not something that I have worked on at all. I know that there are tetraploid cassavas. Actually, Carlos, you probably know more about this than I do.
Yeah, cassava, it seems to be a putative tetraploid, allotetraploid, but nobody certainly knows what are the original species. I mean, there's some suggestions.
I mean, we're basically treating this like a diploid.
As a diploid, yeah.
And we're not worrying about differences in ploidy levels.
Right.
Yeah.
Related to the previous question, what is the best suggestion for dealing with reducing genetic diversity over the cycles?
Right. It is a good question, and it is an issue that people are concerned about precisely because genomic selection allows you to make more cycles than phenotypic selection. So, okay, so I mentioned optimal contributions. That is a good approach. There's evidence that, well, yeah, I mean, I think basically just paying attention. So that is what optimal contributions does. Is it? I mean, and I think breeders have done this sort of non-quantitatively for a while. Once you have a genomic selection population that's underway, that's being improved, it will of course become harder and harder to introgress new genetic diversity into that population, because the new genetic diversity, you know, it has different linkage phases between the markers and the favorable alleles. So any progeny from new diversity that you bring into the program are not going to be very well predicted. So bringing in new diversity is challenging, which isn't to say that it can't be done. In fact, there's a good paper just out in Proceedings of the National Academy of Sciences from Alain Charcosset's group in France, looking at bridging techniques. So you have some new diversity that you want to bring in. You make some bridging crosses with the population that is elite that you're moving along, and then you bring those crosses into the elite population. But yeah, other than that, really just as you're selecting, being willing to go a little bit lower down in your selection criterion, choose individuals that are a little bit lower if they look like they're contributing something valuable from the genetic diversity perspective. That's essentially what optimal contributions does.
All right, now it's time for my question.
Excellent.
Yes. Your genomic selection basically is based on additive effects, or you take into consideration non-additive effects? And how important do you think they are?
Okay, that's a good question. So what matters to me is breeding value. Fundamentally, breeding value is an additive characteristic. Now, there is confounding when you have, so a phenotype, you know, plant breeders always say there's an additive component to the phenotype, a non-additive component, and an error. Well, so when you're just looking at an individual, these three components are somewhat confounded with each other, right? And to the extent that you can sort out what's what, you can get a better estimate of the additive component that really matters to you for selection. Now, okay, so I showed this triangle of the variety development pipeline. The individuals that you want to come out at the end of the pipeline that you're going to propose to farmers as varieties non-additivity is fine for that. And if you have good, I mean, non-additive interactions are harder to predict, okay? But if you have good ways of predicting them, by all means, use that to choose which varieties you're going to move forward toward variety release, or which clones are going to move forward toward variety release. But when it comes to actually choosing parents, I would just rely on the breeding value. So additivity.
Well, thank you so much, Jean-Luc,
Sure.
for your amazing presentation, and also for being so candid responding the questions. Just wanted to take a couple of minutes to announce next week seminar by Dr. Guillaume Bouchet in flax breeding. I think it's going to be an amazingly interesting seminar, and we look forward to it. We have participation today from different places here in the US, Canada, Guatemala, Kenya, Costa Rica, Ethiopia, Nigeria, and Germany, among others. So very good participation. Tomorrow will be an announcement for next week's seminar, and there's going to be also an announcement for a change in speakers for the following seminar on April 20. We'll keep you posted about that, and we'll talk more about that change next week when we meet together again at 11 a.m. Thank you so much for your participation. For those of you that celebrate Easter, have a happy Easter. Have a happy weekend also, and look forward to having you here again next Thursday. Thank you so much, Jean-Luc.
Yeah, you're welcome.
It was very good. Okay, thank you so much. All right.